Welcome everyone to this week's edition of the Extracellular Vesicle Club. I'm very happy to be hosting a team here from Bern, Switzerland, which is actually my ancestral canton in Switzerland. If you go back about 250 years or so, when my ancestors emigrated, and we're going to be hearing about a very nice story with some microRNAs and EVs from Vera Tscherrig. So before I hand it over to you, Vera, I just want to say that everybody can put your comments and questions in the chat box, and we will get to you at the end of the presentation. Then we'll allow you to unmute and show your camera. And I also want to make a note about something that we were just talking about before we started, and that is the MySev guidelines, the minimal information for studies of EVs. Some of you may be co-authors on that document, which started over a year ago and is now approaching completion. So if you are a co-author from last year, please make sure to check your email, check your spam, because we're in the final few days of collecting the co-authors for that manuscript. So please check that out and complete the author survey if you have not. All right, well, Vera, thank you so much for joining today. I'd like to invite you to share your screen and tell us about this very interesting work that you have done.
Okay, thank you very much for that nice introduction, Ken. I hope you're all going to see my screen now. So today I'd like to talk to you about the therapeutic potential of microRNAs from small extracellular vesicles derived from Morton's jelly mesenchymal stromal cells in premature white matter injury. And just to begin to show you all where Ken's ancestors come from, so we are based on Europe, and more precisely, we are based in Switzerland, and there in the capital city in Bern, and here at the Inselspital in Bern. So to briefly introduce what our working group is mainly interested in, we are mainly focused on premature birth. And premature birth is basically any birth that is occurring before the 37th week of gestation. And a normal human term birth usually lasts until like 40 weeks. So any birth that is occurring before that is called a birth too soon. And babies that are born too soon usually have a birth weight that is below 1,500 grams. And worldwide, 1 in 10 of all neonates are born preterm. So it is quite frequent. And it is also the major cause of death in children under the age of 5 years. And it leads to lifelong neurological complications such as visual, hearing, cognitive or motor impairments, as well as mood disorders and also attention deficit disorders. And one of the most common neurological complications upon preterm birth is premature white matter injury. And premature white matter injury is caused by inflammatory and hypoxic ischemic insults. And its clinical presentation is an overall hypomyelination, focal and diffuse white matter lesions, as well as a thinning of the corpus callosum and ventricular enlargement, as you can appreciate here on the right on the illustrated image. And unfortunately, so far no cure exists. Here, I would also like to briefly talk about how the cellular picture looks like in prenatal brain injury compared to healthy brain development. So on the left-hand side, you can see how healthy brain development looks like. There we have homeostatic microglia, which can lead to the maturation of oligodendroglial cells. So they can become from the oligodendrocyte precursor cells to mature oligodendrocytes. And these mature oligodendrocytes are then able to produce the myelin sheets that are surrounding the axons of the neurons that are allowing a normal signal transmission, which leads to healthy cognitive functions. On the other hand, on the right side, you can see in the case of perinatal brain injury, we have hypoxic ischemic or infection inflammatory insults that lead to a pro-inflammatory microglial phenotype. And these pro-inflammatory microglia, they can release inflammatory cytokines, which can lead to reactive astrocytes, but also which can hamper the oligodendroglial maturation. And in this case, we have less mature oligodendrocytes, leading to a lower amount of myelin sheets that are surrounding the axons of the neurons, which in case then results in hypomyelination, leading to neuronal apoptosis, which ends up in cognitive, sensory, and motor deficits, such as most commonly cerebral palsy. And as I told you before, so far there is no cure for white matter injury. However, in the past couple of years, the interest has shifted towards the use of extracellular vesicles, and more specifically of extracellular vesicles from the mesenchymal stromal cells. And in our case, we use mesenchymal stromal cells from the Wharton's jelly, and as almost all cell types, also these Wharton's jelly MSC release different types of extracellular vesicles. And here in our lab, we are distinguishing these extracellular vesicles based on their size, and we are mostly interested in the small type of the extracellular vesicles, so the small EVs. And these small EVs, they contain a different set of molecules such as proteins, DNAs, mRNAs, but also microRNAs, which I will talk about in more detail a bit later on. So in the last couple of years, our research group could have already successfully shown that Wharton's jelly-derived mesenchymal stromal cell-derived small extracellular vesicles are actually able to protect the brain after experimental premature white matter injury. And what they could observe is that when we administer these small extracellular vesicles from the Wharton's jelly MSC intranasally into rat pups that suffer from an experimental premature white matter injury, the results were an improvement of the maturation of the oligodendrocytes, as well as a better myelination of the neurons. But also in the animals, the learning and memory was improved. And on the other hand, the glial activation, the apoptosis, and the overall mortality was reduced. So we then focused a bit more in the last couple of years on why these small extracellular vesicles actually have such a beneficial effect. And we looked into their cargo in a bit more detail. So what we could show is that these microRNAs, based on next generation sequencing analysis, they had these small extracellular vesicles, they have a high amount of microRNAs, highly abundant, compared to the Wharton's jelly MSCs. So all these microRNAs that were highly enriched in the small extracellular vesicles, they were predicted to target pathways that are involved in white matter injury-related processes, such as, for instance, the Notch pathway, but also the MET kinase pathway and TP53. And just more recently, in our recent paper, we could confirm with qPCR that there were these different types of microRNAs highly abundant in the small extracellular vesicles, such as microRNA 22.3p, microRNA 21.5p, microRNA 27b3p, as well as also the members of the LED7 family. And we could show that these highly abundant microRNAs are predicted to target TAUC1 and TP53, which are both involved in inflammation and apoptosis, which are hallmarks of premature white matter injury. So this leads us to our hypothesis that the mature microRNAs contribute to the protective and therapeutic effect that we observe after the administration of Wharton's jelly mesenchymal stromal cell-derived small extracellular vesicles in experimental premature white matter injury. And here, maybe as a quick side note, for those of you who are not so much familiar with what microRNAs are, so microRNAs are basically short non-coding RNAs that are around 19 to 25 nucleotides long, and they are processed in the nucleus with different enzymes, including DROSHA, and they are then further exported into the cytoplasm where they become finally mature, and' with RISC and the help of different proteins such as the organelle proteins, they can efficiently and successfully bind to mRNAs on the 3' untranslated region and then lead to either mRNA degradation or translational repression. As such, we can say microRNAs are able to regulate the gene expression. So now I would also like to tell you a bit how we proceed in order to get to our results and in order to validate the effect of the microRNAs in the small extracellular vesicles. In a first step, we needed to generate naive Wharton's jelly-derived mesenchymal stromal cell-derived small extracellular vesicles. And in order to get to that, we collected the umbilical cords from human term birth from healthy female donors here at the Inselspital in Bern. And we digested the Wharton's jelly mesenchymal stromal cell by enzymatic digestion. And we cultivated these mesenchymal stromal cells until passage 4 to 5. And when the cells were around 80% confluent, we changed the medium and isolated the SEV with differential ultracentrifugation followed by size exclusion chromatography in order to get to a small EV preparation. And on the other hand, in order to be able to compare their microRNA cargo, we did a different preparation for small extracellular vesicles, where we added a silencing RNA for drosha to the Wharton-Shelley mesenchymal stromal cell. And as I just told you before, drosha is important for the maturation process of microRNAs. And when we knock out or knock down drosha in the mesenchymal stromal cells by adding the silencing RNA, we release these mesenchymal stromal cells release small extracellular vesicles with a reduced amount of microRNAs. So we had these two SEV preparations, and we could then compare their effect on experimental premature white matter injury. So with this, I would like to share with you our results that we got so far. In a first step, we wanted to make sure that the SEV preparations that we got with ultracentrifugation followed by size exclusion chromatography are actually meeting the general EV criteria. So we analyzed the vesicles based on their morphology, as you can see here on the picture B, and also based on certain proteins that they are carrying, and also if they are expressing the known tetraspanin CD9, CD81, and CD63. And we also looked into the size and set the potential of these vesicles. And based on that, we could confirm that in our samples, we had an enriched SEV preparation. And as you can appreciate here on the panel E, we also looked into the integrity of the SEV, if they are still able to transfer adenosine to adenosine triphosphate, and we did that with an adenosine fluorescent assay, where we could show that with the administration of our small extracellular vesicles, we could produce a high amount of adenosine triphosphate, indicating that the vesicles were still integrated. And then we looked whether the microRNAs that are carried by these small extracellular vesicles are indeed functional. And in order to get to that, we did a luciferase reporter assay where we had a specific vector that contained the 3' untranslated region of target mRNAs, in this case TP53 and TAOC1, downstream of the luciferase signal. And as I mentioned before, TP53 and also TAOC1, they are relevant for white matter injury as they are involved in apoptosis and inflammation. And we then analyzed the luciferase signal either with the administration of the small extracellular vesicles or without. And in the case of functional microRNA, we would expect to have a reduced light signal, since the microRNAs would able to be bind to the 3' untranslated region of these specific mRNAs and thereby inhibit the luciferase signal. And we could indeed observe that for TAOC1 and also for TP53, where we had a reduced light signal after the administration of our small extracellular vesicles. But we also wanted to make sure that this was not an unspecific effect, and therefore we looked whether the small extracellular vesicles somehow reduce the light signal when there is no specific 3' untranslated region present, and we used therefore a control vector. And in this case, we did not have a reduction of the light signal. And then what we also did, we then modified the binding sites of the 3' untranslated region of TP53 in such a way that the microRNAs that were highly abundant in our small extracellular vesicles, such as LED7 or microRNA 22-3P, could not bind to the 3' untranslated region anymore. And in this case, we observed no reduction of the light signal anymore, indicating that indeed the microRNAs can functionally bind to the target mRNA and result in a light decretion. So with this, we then wanted to compare our microRNA-rich small extracellular vesicles to the drosha knockdown Wharton-Shelley mesenchymal small extracellular vesicles that do not contain a high amount of microRNAs. And in a first step, we needed to confirm that the drosha knockdown indeed resulted in a reduced amount of microRNAs in the extracellular vesicles, and we could observe that there was a significant reduction of almost all of the microRNAs present in our small extracellular vesicles in the naive Wharton-Shelley MSC SEV. And then we also wanted to verify whether these drosha knockdown had an effect on the SEV in such a way as that they would be modified somehow. But when we analyzed their morphology, as well as their size and set potential, and also their protein cargo based on Western blot, we could not observe any changes in the Drosha knockdown SEV, indicating that despite from reducing the amount of microRNAs in the SEV, the Drosha knockdown does not lead to any specific changes in the small extracellular vesicles. So with this, we then used these two preparations of SEV in order to verify whether the microRNAs are functional in different assays related to premature white matter injury. And in the first step, we wanted to see whether the small extracellular vesicles that are enriched, that are high in microRNAs are able to promote oligodendrocyte maturation, and if the Drosha knockdown small extracellular vesicles are able to do the same. And as you can see here, we looked into an oligodendrocyte cell line, the MO313 cells, and we analyzed different gene expression for genes that are known to negatively regulate the oligodendrocyte maturation process. So in this case, this would mean that when we have a reduction of these genes, we have a promotion of oligodendrocyte maturation. And we could show that after 6 and after 24 hours of the addition of our small extracellular vesicles to the MO313 cells, we saw a significant reduction of these genes, indicating that the naive small extracellular vesicles are able to promote the oligodendrocyte maturation. However, in the case of the Drosha knockdown small extracellular vesicles, we did not observe a significant decrease, indicating that only the naive small extracellular vesicles were able to promote oligodendrocyte maturation, and that the microRNAs seem to be relevant for this process. And we then also looked into the anti-apoptotic effect that the small extracellular vesicles have in vitro. And therefore, we did an oxygen and glucose deprivation, and we analyzed after the 6 hours after the oxygen and glucose deprivation whether the extracellular vesicles have an anti-apoptotic effect based on their ability to reduce apoptotic signals such as caspase-3 or BAT. And we could observe that with the administration of naive small extracellular vesicles, the apoptotic markers significantly decreased. However, when we added Drosha knockdown small extracellular vesicles with a reduced amount of microRNAs, this was not the case anymore. And also for the mitochondrial membrane potential, which is another important factor of apoptosis, we saw that when we had oxygen and glucose deprivation, the mitochondrial membrane potential shifted towards an unhealthy mitochondria that had a higher amount of JC1 monomers, indicated here in green signal with the JC1 assay that we used. And when we had healthy mitochondria, this would then result in a higher amount of red signal of JC1 aggregates. And only when we added naive small extracellular vesicles after the oxygen and glucose deprivation, we were able to observe a shift towards these healthy mitochondria by an increase in the red signal. And this was not the case anymore when we had the Drosha knockdown small extracellular vesicles added to the cells. So also here it indicates that indeed the microRNAs are relevant for the anti-apoptotic potential in vitro. So this leads me to our first conclusion, where we can say that in vitro we have observed that the microRNAs play a key role for their anti-apoptotic effect, but also for their effects to promote oligodendrocyte maturation. And now in the last couple of months, we wanted to check these in vitro data further and verify this also in an in vivo model. And we would like to publish this data shortly, and therefore today I'm only able to show you some illustrative data of what we got so far in order to protect our results that we have so far. So what we did is actually we used an already established animal model of premature white matter injury, where we added an inflammation due to intraperitoneal administration of LPS, followed by a carotis cauterization in order to induce an ischemic insult, and then followed by a hypoxia of red pups at P2 in order to induce to them premature white matter injury. And 24 hours after we induce the insult to these red pups, we add there either naive small extracellular vesicles or the Drosha knockdown small extracellular vesicles. And what we also did is we added DIR labeled small extracellular vesicles in order to verify the SEV biodistribution and their kinetics. So following 24 hours after the administration of the small extracellular vesicles, we did our analysis where we looked into biodistribution, but also on the effect of the microRNAs on myelination and neuroinflammation. And what we got so far is, I'm sorry that I only show you illustrative data, but we could observe that when we add the DIR labeled small extracellular vesicles intranasally to our red pups, we measured a signal of DIR dye in the brain already 1 hour after the administration, and also after 6 hours, as well as after 24 hours, the signal remained present in the brain. However, what we also observed based on fluorescent imaging was that we had a high signal in the belly area. And in order to verify where this signal is actually coming from, we did then also an ex vivo analysis of the different organs, such as the lung, the liver, the spleen, and the digestive tract. And we could actually verify that a high amount of the SEV signal is also going into the digestive tract, which is not so much unsurprisingly, because a high amount of the small extracellular vesicles that you administer intranasally will be swallowed by the pups and will be decreased out of the system through the digestive tract. And then what we also did is we analyzed the brains based on their ability to reduce microglia activation and promote oligodendrocyte maturation of the small extracellular vesicles. So we had 4 different groups of animals. We had a control animal that did not suffer from premature white matter injury. And then we had animals that were injured and that received a vehicle only, so PBS only. And then we had the 2 EV preparation, once the naive small extracellular vesicles with a high amount of microRNAs, and on the other hand, the Drosha knockdown small extracellular vesicles intranasally administered to the red pups. And we analyzed the brains based on their microglial activation. And as you can see here, when we have a healthy brain development, we have these homeostatic microglia that are basically have a morphology with these branched shapes. And then when we have the prenatal brain injury, we have the infection, the hypoxia that leads to a more amoeboid morphology of the microRNAs. And also these microRNAs express high amount of IBA1 and CD68. And what we were able to observe is that indeed, when we have our injured pups that received vehicle only, we have a high increase of IBA1 and CD68. But when we add this naive small extracellular vesicles, we were able to show that they were able to reduce the IBA1 signal significantly, not only on immunohistochemistry slides, but also on gene expression levels and protein expression levels, which was not the case for the Drosha knockdown small extracellular vesicles. However, interestingly, for the case for CD68, we observed also an effect, even with the Drosha knockdown small extracellular vesicles, indicating that for this regulation, the microRNAs alone might not be the only contributor to a beneficial effect. And we then also analyzed the brains at P11, so postnatal day 11, in order to look into the maturation of the oligodendrocytes. And when oligodendrocytes become mature, they express myelin basic protein, or MBP in short. And this MBP you can stain for MBP with different techniques. And we were able to analyze the MBP signal, and we could show that with the injured pups, we have a highly reduction of MBP signal, which was also the case even though when we added the Drosha knockdown small extracellular vesicles. However, the able to somehow increase the amount of MBP, and as such we can say that the microRNAs are relevant for the MBP, for the oligodendrocyte maturation. And with this, I like to conclude that our results strongly indicate the critical relevance of the microRNA cargo and the therapeutic effect of Wharton's-Jelly MSC SEV in premature white matter injury. And intranasal administration seems to be a minimally invasive method to deliver the Wharton's-Jelly MSC SEV to the brain. And so what comes next? We would like to potentially go with these small extracellular vesicles that show so many beneficial effects to the clinics, and to hopefully one day have a minimally invasive administration technique with using an with using a nasal spray for the preterm born babies in order to allow them to have the same life qualities as their term born peers. And with this, I'd like to thank you all very much for your attention. And also I'd like to thank everyone that contributed to this work, and to you, Ken, for inviting us here to present our data. Thank you very much.
Fantastic. Well, thank you for sharing. And it's time now to have our question and answer session. So I have allowed people to unmute themselves now and to show their camera if they are ready to do that. So I will go through the chat box, and when I call on you, please be ready to deliver your question. All right, so let's get started with Norm Howie. Norm, you have a couple of questions, so please go ahead and ask them both.
I do. Thank you for that really neat work. My one technical question, and then two sort of more curiosity questions. And I guess the technical question is curiosity too. I'm always curious why people isolate one method versus another. So a lot of times people will use columns, let's say with plasma or something, to clean it up and then do ultracentrifugation. In your case, you did the opposite. And I was just curious what the rationale is for running a column after the ultra.
Well, we basically do it also because we have quite a high starting material. We actually, so far, we culture our Wharton's-Jelly MSCs in the usual 2D flasks. So we have around 200 ml that we start with. And the ultracentrifugation is also kind of like allowing us to concentrate the volume a little bit, that we then can put it on a column and purify it further. That's a bit why we do that, yeah.
Got it. And the last two are sort of related. Do you plan to conduct some ultrastructural studies? So one thing is maturation of oligodendrocytes, and of course the other thing would be how well they actually wrap the G ratio around an axon. Is that sort of in the plans before publication? I see somebody nodding.
Well, that's actually a very great thing that one should really look into, but we did not plan so far to look into the wrapping around the axons. It's supposed to be probably quite technically, and yeah, what would you suggest in order to analyze the wrapping around the axons?
It's actually not that bad. It's electron microscopy and then simple measurements after that. So okay. The electron microscopy is, I don't know how to do it. We get somebody else to do it, but the rest is pretty straightforward. And then the last thing was sort of related to that. I'd love to see this in the Cooper zone model for similar reasons. So in the Cooper zone model, you have a posterior demyelination, and then it remyelinates, and it doesn't remyelinate right. It's off. And there's possible reasons for that. If this could intervene in that process and sort of get a better wrapping, that would be very interesting.
That would be very interesting indeed. Yeah, that's something we should really consider looking into. Thank you for bringing that up.
If you are interested, I'm happy to introduce you to Peter Calabrese. He's one of the guys at Hopkins who does a lot of MS work, and he knows that model very well.
Yeah, that would be lovely, actually. Thank you. Yeah, thank you.
Thank you.
And I see there's a follow-up here, Kathleen Borgman. Did you want to mention what you responded to Norm's comment?
Hi, I would just love to see similar studies. I'm a program officer at NIDA. I would love to see similar studies looking at whether or not it can be productive following illicit drug or alcohol exposure. It could easily be translated into therapeutic model for infants that are born in that capacity. And this study is really interesting. Good work.
Thank you. And that's actually a really good point too. Yeah, if you could use it for different models of disease, that would be something that would be really great one day. Yeah.
Yeah, thanks for the comment. Great idea. Phil Askanese.
Hi, this is wonderful work, beautifully presented, wonderfully illustrated.
Thank you.
And we are considering some neuro models and immunological models where the sort of primary EVs influence local macrophage, or in this case, microglia cells, and then they make secondary EVs to give the effects measured. So I don't know if you're able to consider that sort of an EV chain rather than direct effect of the Wharton's jelly EVs on the oligodendrocytes.
Yeah, that's actually a very good point. And I think this could also very likely be what is happening in the brain. And the unfortunate thing is that until now, we do not really understand how the EVs influence these cells. So it could actually very much be that they are taken up by the microglial cells, and that they are not only allowing them to become reducedly activated, but also allowing them to extrude extracellular vesicles that, on the other hand, have enriched therapeutic potential that then goes to other cells. And what would also be interesting is to see whether this then also goes into the systemic, that it is probably not only remaining in the brain and in the brain cells, but maybe then we have enriched amount of therapeutic EVs that could lead to an improved phenotype of the outcome of the disease. Indeed, yeah.
Well, you got exactly what I said and what I meant.
Yeah.
And this kind of thinking overcomes some difficulties that EVs will work across MHC boundaries, that they seem to have no disease specificity themselves, but the disease specificity may be in these ancillary cells at the site. And then there's the big problem of not finding enough miRNAs to account for biological effects, and they could be provided by the intermediary cells. Wonderful, wonderful. I wonder if I could go to BioVendor and get copies of your figures, which I love.
Yeah, I can recommend it. Yeah.
Thank you very much.
Thank you.
Thanks, Phil. Next, we have a question from Sujata Mohanty. Sujata, please go ahead.
Hi, Ken. Thank you so much. Fantastic work. Yeah. So my question is, like, you have used the intranasal route. So what about intraperitoneal or intravenous? Will there be difference in the delivery of small extracellular vesicle to the brain in terms of amount or time? Because if they are very premature, would intranasal will be the better option vis-a-vis other route of administration? Thank you.
Thank you very much for that question. We did not do different administration routes ourselves, but it has been shown that when you use the intravenous or the intraperitoneal injection method, in that case, the EVs have really to cross the brain barrier in order to reach the brain. And they are also the first barrier that they got, or the first direction that they're targeting to is quite often the liver. So they're very often enriched in the liver and not really able to target to the brain. So therefore, I think the intranasal administration is, on one hand, a very efficient method to target EVs to the brain. And on the other hand, it also allows you to kind of like bypass the blood-brain barrier in order to reach the brain through different mechanisms. And I also think that even if the neonates are born very prematurely, I still think that the intranasal administration is less invasive. And also when you kind of like consider what we did not do yet is actually to find out the right timing and dosing, and if multiple doses are required. And let's say you need to apply many doses, or let's say even just 5 a day, it would be quite more handy for parents to be able to do it themselves at home. And I think that the intranasal administration would allow a very easy administration technique that maybe the parents could learn and also administer themselves. While as on the other hand, the intravenous and also the intraperitoneal administration would not be so handy for parents themselves. And as such, I would kind of like recommend to investigate further in the intranasal administration of the SEV in the case of the premature born neonates.
Perfect. Thank you so much. Yeah,
Thank you
I got
too.
it. Thank you, dear.
Just as a kind of follow-up on that, you know, in some of the rodent models, there's a pre-treatment that's done of the nasal cavity. Do you do a pre-treatment? I might have missed that.
Yeah, I'm sorry, I did not mention it here, but yeah, actually we do a pre-treatment. We add 30 minutes before we add the SEV hyaluronidase.
Oh, I can't say that. I can't say that one. I'm not even going to try. Thank you for doing it.
But we add that, yeah, indeed. Yeah, that's also true. That's maybe something that you would not be able to do in the human case.
Mm-hmm.
Or that would not be as convenient as that to do as well. That's another thing that we should consider when we think about translating it to the clinics.
So I would like to also just switch a little bit the topic and ask you more about the microRNAs. What form do you have evidence of what form the microRNAs are in your EVs? Are they you know, in a precursor form? Are they complex with a protein? Is that something that you've given thought to?
No, actually not. We didn't really investigate that further yet, but it could be quite likely, since how we get rid of most of them is by doing the knockdown of the drosha, which is quite early on the maturation step. So it could still be that a lot of the microRNAs that are present in the naive SEV are still in these precursor forms, and that they are present in that form. But we did not really look in detail into at what stage they are of the maturation. What we see is we also have a different amount, like we have 22.3p, but on the other hand, we have 21.5p that is quite highly abundant. So they are both like the 3p and the 5p types of the microRNAs. So that would be something that we should follow up in more detail in the next time. Yeah.
Yeah, lots of interesting questions that always come out of results. So it looks like we've come to the end of the questions that are in the chat box. So if anybody has a last minute question, please put it in there or raise your hand quickly. Otherwise, I don't know. Mariana, Andrina, did you want to say anything here at the end? Oh, we do have one question. Yes, indeed. Please go ahead. We have two. All right, we have two. So let's see, Shilpa, and then Kathleen.
Great talk. Really enjoyed it, Vera. So have you done the sequencing to find out what percentage of microRNAs are present in these EVs? And do you actually see the viRNA? Because they're gaining a lot of momentum now in terms of their therapeutic benefits. So I was just wondering if you've seen the viRNAs.
So what we did is we did next generation sequencing, and we then just looked into the microRNAs that had a hit that was higher than 10,000 compared to the Wharton-Shelley MSCs themselves. So that's how we looked into the amount of how highly they were enriched. And in terms of the RNAs, we did not look into that yet, but that would definitely also that would be very interesting to consider looking at with next generation sequencing analysis of the EV preps. Yeah, indeed.
Great.
Thank you.
Good work.
Thank you.
And Kathleen?
I was just wondering that since you have a signal in the gut that EVs are also accumulating there, if you've considered mechanisms of the gut-brain axis in the mechanisms that are altering the white matter changes that you're seeing.
Yeah, I'm glad you asked. Thank you very much. Because also actually in the case of the premature born, it actually also is another disease that happens quite frequently, is that they have an inflamed gut, or that they have the irritated bowel disease. They're quite they are more likely to have it compared to healthy term-born peers. And that's also something that could be the case why the SEVs are actually then traveling into the gut, because there's also an inflammatory signal that is highly there, and maybe the EVs are attracted due to this inflammatory signal, and then go down through the microbiota gut-brain barrier And yeah, that's also one thing. And I think it also has been shown that the microRNAs could also go through this axis, and that would be maybe another thing that would be quite interesting to investigate further. Yeah. Thank you.
Well, you're injecting LPS perinatally, right? And so have you guys looked at any of the damage that might be occurring there and whether or not the EVs are protecting or changing that inflammation in the gut?
No, so far we only looked into the brain damage, but that would actually really be something very nice to investigate in further. Yeah, especially if you see now that the EVs are traveling there. Yeah, they could have a therapeutic potential there as well. Yeah.
Very good. All right. Well, if there are no more questions, then I can only echo what Phil had to say, which is that it was a very beautifully presented piece of work here today. So thank you very much, Vera, for joining, and thanks everybody for contributing with questions. And we'll hope to see you all on an EV Club coming up soon. Take care, everybody.
Thank you. Bye. Thank you.
Bye.